This presentation from Natural Scientifics examines manic depressive illness, commonly referred to as bipolar spectrum disorder, through a systems biology perspective. The scientific framework and visual architecture were developed by Brian Geniuson, research scientist, as part of ongoing research at Natural Scientific's trademark. Bipolar disorder is traditionally described as a mood disorder characterized by alternating episodes of mania and depression. However, converging evidence indicates that this condition is more accurately understood as a state-switching neural instability syndrome, emerging from interacting failures across energy metabolism, neurotransmitter network regulation, immune signaling, circadian timing, and stress adaptation. Rather than reflecting isolated chemical imbalances, manic and depressive states arise when core stabilizing systems of the brain lose coordination, resulting in oscillatory instability across neural circuits. At the center of this model is oscillating instability between manic, depressive, and mixed affective states. These states do not occur randomly. They reflect predictable biological transitions driven by system-level vulnerability. The first destabilizing domain is neural energy metabolism. Neurons associated with bipolar disorder consistently demonstrate mitochondrial inefficiency, impaired ATP production, disrupted redox balance, and elevated oxidative stress. During manic states, neural circuits enter hypermetabolic inefficiency, characterized by increased activity with poor energetic yield. During depressive states, this instability collapses into cellular energy deficit, reducing synaptic firing capacity and cognitive drive. Energy instability lowers the threshold for state transitions across the brain. Bipolar disorder is not a monoamine deficiency. Instead, it reflects a failure of network-level neurotransmitter coordination. Dopaminergic signaling displays amplitude instability, glutamatergic excitation overwhelms inhibitory GABA control, and serotonergic systems function primarily as secondary modulators. The critical failure is not chemical availability, but signal integration and temporal regulation across neural networks. Another destabilizing layer is chronic neuroinflammation. Microglia, the brain's immune synaptic regulators, become persistently primed. When overactivated, they disrupt synaptic pruning, alter connectivity patterns, and amplify excitatory signaling. Inflammation does not initiate bipolar disorder, but it accelerates phase switching, increases episode severity, and contributes to long-term neuroprogression. Circadian regulation represents a causal driver rather than a downstream symptom. Instability in core clock gene signaling disrupts sleep-wake timing and alters epigenetic regulation of neuronal excitability. Sleep deprivation therefore represents one of the most potent biological triggers of manic episodes due to destabilization of neural timing systems that normally constrain excitation. Bipolar disorder also involves dysfunction of stress recovery systems, flattened cortisol rhythms, impaired feedback inhibition, and cumulative allostatic load reduce the brain's capacity to stabilize following perturbation. Under these conditions, stress becomes a destabilizing biological force rather than an adaptive signal. When energy instability, network volatility, inflammation, circadian disruption, and stress dysregulation interact, the brain enters a condition where minor perturbations produce large-scale state transitions. This reframes bipolar disorder as a disorder of neural systems integration rather than mood imbalance alone. Human clinical and translational studies consistently demonstrate altered cerebral energy metabolism, circadian rhythm abnormalities preceding mood episodes, elevated inflammatory markers, and stress axis dysregulation across bipolar phenotypes. Mechanistic and preclinical evidence further reveals mitochondrial dysfunction, microglial-mediated synaptic remodeling, circadian gene disruption, and amplification of network instability under stress. Together, these findings support a coherent biological explanation for oscillatory mood states. This systems-level framework supports mechanism-matched intervention strategies, including energy stabilization, modulation of neuroinflammation, circadian reentrainment, synaptic network stabilization, and stress response recalibration. Such approaches move beyond symptom suppression toward biological stabilization. Full supporting scientific literature is available separately in the video description. This content is provided for educational and scientific discussion purposes only. It does not constitute medical advice, diagnosis, or treatment recommendations. Copyright 2026 Natural Scientifics. Scientific Architecture by Brian Geniuson. Research Scientist. Educational content only. No medical advice.